Good morning, students. Our today's topic is chemical reactions. Chemical compounds undergo two types of changes, a physical change. This simply refers to a change in shape without breaking of bonds. This is a physical process. Another physical change is a change in state of matter. Example is ice melts into water or when water becomes a vapor. These are also physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. A rate of a physical or chemical process refers to the amount of product formed per unit time. It can be represented as rate is equal to delta P upon delta T. Rate of physical and chemical processes are influenced by temperature among other factors. A general rule is that rate doubles or decreases by half for every 10 degree centigrade change in either direction. Catalyzed reactions proceed at a rates higher than that of uncatalyzed ones. So, in which catalyst is present, that reaction occurs at a very fast rate. When enzyme catalyzed reactions are observed, the rate would be higher than the same but uncatalyzed reactions. For example, when carbon dioxide plus water forms carbonic acid in the presence of carbonic anhydrase. Carbonic anhydrase is the enzyme which fastens this reaction. In the absence of any enzyme, this reaction is very slow, with about 200 molecules of carbonic acid being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6 lakh molecules being formed every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzyme is incredible indeed. So when enzyme is present, the reaction proceeds at a very fast rate. A multi-step reaction, when each of the step is catalyzed by the same enzyme complex or different enzymes, is called a metabolic pathway. For example, glucose changes into pyruvic acid is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzyme catalyzed metabolic reactions. You should know that this very metabolic pathway with one or two additional reactions give rise to a variety of metabolic end products. In our skeletal muscle, under anaerobic conditions, lactic acid is formed. Under normal aerobic conditions, pyruvic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol, that is ethyl alcohol. Hence, in different conditions, different products are possible. That you have studied in class 10th also, the different ways of breakdown of glucose. Under aerobic conditions, pyruvic acid changes into carbon dioxide and water. Under anaerobic conditions, pyruvic acid changes into ethanol and carbon dioxide. And in the lack of oxygen in muscle, it changes into lactic acid. How do enzymes bring about such high rate of chemical conversions? The chemical or metabolic conversion refers to a reaction. The chemical which is converted into product is called a substrate. Hence, enzymes, that is proteins with three-dimensional structure including an active site, convert a substrate into a product symbolically as changes into P. So under the reaction, substrate changes into product in the presence of an enzyme. Substrate S has to bind the enzyme at its active site within a given cleft or pocket. First, the substrate fixes itself in the active site of the enzyme. The substrate has to diffuse towards the active site. Thus, there is a obligatory formation of an enzyme substrate complex, ES complex, where E stands for enzyme. The complex formation is a transit phenomenon. During the state where substrate is bound to the enzyme active site, a new structure of the substrate called transition state structure is formed. When the substrate fixes itself in the active site of the enzyme, a transition state is formed. Very soon after the expected bond breaking or making is completed, the product is released from the active site. In other words, the structure of substrate gets transformed into the structure of product. So क्या हुआ, सबसे पहले substrate एंजाइम की एक्टिव साइट पे जाके कंबाइन करेगा, एंजाइम सबस्ट्रेट कॉम्प्लेक्स बनेगा, इसमें कुछ बॉन्ड फॉर्म होंगे, कुछ बॉन्ड्स ब्रेक होंगे और एंड में एक प्रोडक्ट बनेगा और उस प्रोडक्ट को उस एक्टिव साइट से रिलीज़ कर दिया जाएगा और एंजाइम फिर वेकेंट हो गया। उस एंजाइम की एक्टिव साइट पे अब कोई और दूसरा सबस्ट्रेट का मॉलिक्यूल आके ज्वाइन कर जाएगा। इस तरह से रिएक्शन प्रोसीड करती है। इन अदर वर्ड्स, the pathway of this transformation must go through the so-called transition state structure. There could be many more altered structural sites between the stable state and the product. Implicit in this statement is the fact that all other intermediate structures are unstable. Between the substrate and the product, many unstable structures are formed. Stability is somehow related to the energy status of the molecule or the structure. The y-axis represent in the above graph, the y-axis represents the potential energy content. The x-axis represents the progression of the structural transformation or states through the transition state. The energy level difference between S and P. First, substrate is there. Just see this figure 9.6. Here substrate is there. It goes into the transition state by getting some amount of energy. This is activation energy. When enzyme is present, that activation energy is less, and without enzyme, activation energy is more. So after transition state, the product is formed. The product has the lower potential energy as compared to the substrate, which gives it more stability. So if P is at lower level than S, the reaction is an exothermic reaction. One need not supply energy in order to form the product. In the exothermic reaction, heat is released. However, whether it is an exothermic or endothermic or spontaneous reaction or endothermic or energy requiring reaction, the S has to go through a much higher energy state or transition state. Whether it is exothermic reaction or endothermic reaction, one transition state is formed. The difference in average energy content of S from that of this transition state is called activation energy. What is activation? At transition state some energy is there, and at the substrate some energy is there. The difference between the transition energy of the transition state and the substrate energy is known as activation energy. When enzyme is present, activation energy is less, and without enzyme, activation energy is more. Next is nature of enzyme action. Each enzyme has a substrate binding site in its molecule, so that a high reactive energy substrate complex is produced. This complex is short-lived and dissociates into its product, and the unchanged enzyme with an intermediate formation of the enzyme product EP. The formation of the ES complex is essential for catalysis. So in the reaction below they have shown first enzyme plus substrate forms ES, that is enzyme substrate complex. Then some bonds are formed, some are broken, and it forms enzyme product complex. Then product is released and enzyme becomes free. The catalytic cycle of an enzyme action can be described as: first, the substrate binds to the active site of the enzyme, fitting it into the active site. Second point, the binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. Next point is the active site of the enzyme now is in close proximity of the substrate, breaks the chemical bonds of the substrate, and the new enzyme product complex is formed. Fourth point is the enzyme releases the products of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. I hope this is clear. Next topic, that is factors affecting enzyme activity, we will study next time. Thank you.